This video will talk about the function of T cells, cytotoxic T cells, and B cells, antibody-producing cells, as part of the acquired immune system. So the second important piece is B cell activation. And for B cell activation, we have a very similar process, except that the end result is going to be antibodies. So there are gene rearrangements of this gene, variation in the antibody gene. Activation of the B cell again requires these double events, and the combination cause mitosis and developing after multiple divisions into active cells that secrete antibodies that will bind to free bacteria or viruses. These are B cells. Notice that the receptor shape is different. We have two binding pockets on each. Each of the binding pockets on that receptor is a duplicate, and we have a subset of the different combinations. In the case of a B cell, the event has to be some molecule from a bacterium or a virus. It can be a secreted free molecule, or it could be actually bound to the surface of the bacterium and virus. I've not shown these to scale. In fact, on this scale, we wouldn't even see the receptor, and a bacterium would only be about this big. The B cell moves this part of the bacterium into the cell. If it's free, it simply moves it in and puts it into a binding protein. That binding protein then gets moved to the surface. Remember, this is the fragment of bacterium or virus that was in the extracellular fluid. That binding again to a matching helper T cell. So that helper T cell also has to bind the same fragment held in an MHC2 again. And when binding happens, that activates a second messenger cascade and results in cytokine release. The cytokines bind to a receptor on the B cell, and that activates a second messenger cascade that causes cell division. So we had two events. We had the molecule from the bacterium or virus, and two, a cytokine from a helper T, and again that's a matching helper T that can also bind. So we've got one, bacterium or virus binding, and two, the cytokine. That leads to cell division. Again, notice we have exactly the same A3B1C2, 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 and those now start producing antibody. The B cell receptor is actually the same as the antibody, except the B cell receptor has a portion of the antibody that holds it in the membrane. The antibodies are free and they're secreted from the activated B cell. The important part is they have the same binding pocket, so they also will bind this fragment. That could be a toxin produced by the bacterium, it could be some other product of the bacterium, or it could be a surface protein of the bacterium. These free antibodies are released into the extracellular fluid in the blood, and wherever they bump into that molecule on a bacterium, they bind. Bound antibody binds to receptors on macrophages, and those receptors, once they bind on the macrophage, wind up making this macrophage engulf the bacterium and fuse it with a lysosome, which kills and digests the bacterium. Bound antibody attached to a target moves that into a macrophage for destruction. A second function of antibodies, because of those two arms, the same antibody can actually bind two different bacteria. And by doing so, they clump bacteria into a tight mass. These are not to scale. These proteins, of course, are much smaller than the bacterium. And so the bacteria are held tightly together, which starves them and prevents normal replication, and of course marks them for macrophages. And again, as with T cells, at the end there's a series of cell divisions that results in memory B cells, again with exactly the same A3B1C2. And these memory cells have the antibody as a surface B cell receptor ready and waiting for a new infection. So to run through the whole thing, we have stem cells that during development go through multiple rounds of cell division to produce inactive, immature B cells. At this stage, with both T cells and B cells, any cell that could bind a self cell, anything that has receptors that will bind self molecules, get killed. And so the only ones left are the ones that don't bind to anything that's normally free in the body and should bind only to invaders. When there's an infection, a bacterium, molecules from that bacterium bind to a B cell receptor. That gets internalized and put into an MHC class II protein, which gets moved to the surface. And if a helper T cell has a receptor that matches it perfectly, it'll release cytokines that then activate the B cell. That causes multiple rounds of cell division and active plasma B cells. The plasma comes because they're secreting components of the plasma, antibodies, that clump bacteria or viruses and cause them to be ingested by macrophages for destruction. At the end of the infection, there are multiple memory B cells that have those same binding pockets, and now there are many of them. It's a much faster response to a new infection.